On behalf of the U.S. Women in Nuclear Professional Development Committee, thank you for joining us today. I'm Tamara Baker, the U.S. Wind Professional Development Chair, and I work at the Palo Verde Generating Station in Nuclear Policy and Strategic Business Planning. We are recording this webinar, and it will be uploaded to the U.S. Wind Professional Development Library, so please share it with your chapters. Every year, March is designated as Women's History Month to honor women's contributions in American history. This year's theme is Celebrating the Women Who Tell Our Stories. To celebrate the contributions and achievements of women in the nuclear field, U.S. Wind has crafted a series of professional development events. Maria Lacow, who is the featured U.S. Wind member spotlight this month, kicked off Women's History Month and shared her story about the importance of building your personal board of directors. Tara Nider and Carol Lane shared their stories about first-of-a-kind reactor technology designs and working with the Department of Energy and Congress to advance its innovative technology. And today, we are closing out Women's History Month with Julie Ezold and Sandra Davern and celebrating their remarkable stories about the advanced science of radioisotopes. So with that, please allow me to introduce our speakers and moderator. Our first speaker today is Julie Ezold. Julie has more than 30 years experience in nuclear sciences and is currently the Californium 252 Program Manager at Oak Ridge National Laboratory, or ORNL. ORNL is a multi-million-dollar enterprise that includes the production of berkelium-249, Einsteinium-253, and other transuranium products that have been used in the discovery of a new element 117, which is aptly named Tennessean. Prior to her current position, Julie was the radioisotope production and operations section head, and also the independent oversight manager at ORNL. She also worked as the nuclear criticality safety engineer at the Y-12 National Security Complex and was a shift technical advisor for enriched uranium operations. Julie holds a master's degree in nuclear engineering from North Carolina State University and a Bachelor of Science degree in nuclear energy from Rensselaer Polytechnic Institute. She's been a member of the American Nuclear Society since 1988, where she presently serves on the board of directors and as chairperson of the Scholarship Policy and Coordination Committee. She's also been actively involved with U.S. Wind since 2004, where she currently serves in several leadership roles, including U.S. Wind Steering Committee member and Region 2 executive sponsor. She is also a member of the U.S. Wind Communications Committee. Our second speaker is Sandra Davern. Sandra is the section head for radioisotope research and development at Oak Ridge National Laboratory. She is also the lead for accelerating radiotherapeutics through Advanced Molecular Constructs Initiative. Sandra joined ORNL in 2015 as a radioisotope researcher. Prior to that, she worked at Excel Engineering as an environmental scientist for the Department of Energy's Plant Radiochemistry and Radioimaging scientific focus area. Sandra holds a doctoral degree in cell biology from University College Dublin in Ireland and a Bachelor of Science degree in biotechnology from Dublin City University. She is a member of a number of professional organizations, including the ORNL Women in Physical Sciences, ORNL Women in Nuclear and Global Security, Oak Ridge Women in Nuclear, Society for Nuclear Medicine and Molecular Imaging, American Association for the Advancement of Science, and the American Chemical Society. Our moderator today is Stephanie, who goes by Stevie Dar. Stevie is a senior level project manager with over 20 years experience in the energy industry. She recently joined GSC Solutions as an engineering project manager. Prior to GSC Solutions, she was a senior site project manager at Davis Besse. She holds a Master of Business Administration and Project Management, Bachelor of Science in Mechanical Engineering Design and Industrial Electricity, and a yellow belt in Lean Six Sigma. She is also the co-chair of the U.S. Women in Nuclear Professional Development Strengthened Subcommittee. Please help me in welcoming our speakers and moderator before I turn this over to Stevie.
Okay, thank you so much, Tamara. Good afternoon, everyone. Thank you for joining us for the final session of our 2023 technical webinar series. As Tamara mentioned, my name is Stevie, and I'm a co-chair of the Professional Development Strengthened Subcommittee. I'd like to go over a few logistics before we begin today. So today's webinar will be 90 minutes long. The first part of the session will include 20 to 30-minute presentations from each of our speakers, after which we will take questions from the audience. So for the Q&A portion, you can either type your question into the chat box, or if you prefer to ask your question aloud, you can use the raise hand feature and our host will unmute you. You're not going to be able to unmute yourself, so please wait for the host to unmute you. And then after that, I will call on you to ask your question. We'll try to get through as many questions as we possibly can. And then after the Q&A session, the committee chair will then take a few minutes to close out our session. And now let's turn it over to Julie Ezold. I believe you're muted, Julie.
Okay, I'm trying to get my screens to work. Hold on just one moment here. It worked a moment ago. Okay, are you seeing the presentation?
Yes,
Okay.
we are.
Okay, great. So good afternoon or morning, depending on where you are. Thank you all so much for this invitation. This is my favorite topic to talk about. Today, I'm going to cover industrial and super heavy element research that we're doing at Oak Ridge National Lab, most of which is going to be more in the production side of things. So we'll do a little bit of history. We'll go into the industrial isotopes and then where we are with the super heavy elements. So if you're not familiar, Oak Ridge National Lab is located in Tennessee. We're in East Tennessee, near the town of Oak Ridge. We're about 20 miles from Knoxville. We have really more of the campuses, but two main campuses. The one on your left, the Mountain Valley, that is where our high flux isotope reactor and our radiochemical engineering development center are. And then on the right, we have our Bethel Valley campus, where we have several hot cell facilities and multiple research facilities. So isotope production actually started at Oak Ridge National Lab in 1946. So we were part of the Manhattan Project and then quickly went into isotope production as well as other areas of research. So kind of a little bit of a timeline. 1946, we made our first shipment to Bernard Hospital in St. Louis. It was carbon-14 used for skin cancer. And then during the 1960s, we were making over 100,000 shipments of radioisotopes annually. So creating quite a bit of radioisotopes. In 1966, the high flux isotope reactor began its operation, and so did the Radiochemical Engineering Development Center hot cell facilities. And we'll talk more details of those as we go through the presentation. And then through the 70s to today, we're focusing more on specialty isotopes, both research and production. And these are going to be more for isotopes that cannot be made any other place, specifically with industry. And then, of course, I'm going to highlight the berkelium-249 that was used in the discovery of element 117, which we did get to name as Tennessee. So what makes us unique is both our facilities and our capabilities at Oak Ridge National Laboratory. So the high flux isotope reactor, it is a very unique reactor in the sense that it runs at 85 megawatts, which is very high, almost by a factor of 10 for a research reactor, but it's also greater than a factor of 10 less than a commercial nuclear power. So it's kind of this in between, but you're going to see what really truly makes it unique for isotope production is the thermal neutron flux. At its maximum, it is 2.1 times 10 to the 15 neutrons per centimeter squared per second. So an extremely high thermal neutron flux. We also have a decent or fairly high epithermal as well as fast neutron flux. Adjacent to the reactor is a beamline that is used as a cold neutron source. And what that means is we actually slow those neutrons down to a point that gravity actually affects the neutrons. So if you can't even wrap your mind around that, but what that does is it gives it the term brightest cold neutrons, and it allows the neutrons to penetrate and do imaging at a scale that's never been done before. The high flux isotope reactor operates about 6 to 7 cycles per year, where a cycle is about 23 days before we have to change out the fuel element. And in the upper right picture, what's being lifted out, that is a fuel element. And then if you look at the lower left picture, you'll see kind of this inner and then outer curve shape. Those curve shapes are the fuel plates that are welded. You have an inner fuel element, an outer fuel element. And that whole entire assembly, and it's a joke, but it's about the size of a pony keg, to give you a field of reference for it. The missions of the high flux isotope reactor, as you can see, are neutron scattering, of course, isotopes, because that was its initial, that's what the I stands for, and then materials research, and then the neutron activation analysis. Some of our hot cell facilities, building 4501 is on that Bethel Valley campus, or our main campus, has 4 hot cells, and you can see them depicted on this slide. And we try to keep them for individual radioisotope processing, but because we handle so many different isotopes, sometimes we're going to have to decontaminate the cell to do the next isotope. Cell B is unique. It's the one right here in this corner. That is where we can do the transloading of bringing in the irradiated samples, and then we move them to the different cells for their processing. In this hot cell facility, we can do the radium lead generators, the nickel 63, which I'll go into more detail later on, tungsten 99 dispensing, and then also high purity thorium 229 dispensing. Building 3047 is also on our Bethel Valley campus. It has 4 hot cells, but it is primarily dedicated to the irradiation of making the pellets of radium-226, which is then irradiated in the reactor and brought back for the processing of the actinium-227, which I believe Sandra is going to go into more detail on actinium-227. But it is a dedicated facility for that effort. The Radiochemical Engineering Development Center, as I mentioned, is in the Melton Valley campus. It is adjacent to the high flux isotope reactor. It is actually 2 hot cell facilities. Building 7920 is depicted on the left of your screen, and building 7930 is depicted on the right. And you can see building 7920 has a total of 10 shielded hot cells, 2 shielded caves, 9 glove boxes. Of those glove box labs, 5 glove box labs are dedicated to alpha producing radioisotopes. We have 2 chemical labs or cold labs for doing our reagent makeup and tracer testing, and 2 analytical labs that allow us to do our radiochemical analysis as well as get our isotopics. We have fume hoods. And then in building 7930, the one on the right, that is shaped like a T with the cells, cell A, B, and then C, D, E, and G. Cell F is actually below. We didn't stop it, but it's in the basement. There is where we predominantly do our californium-252 wire fabrication. So give you a sense of the portfolio that we have for radioisotope production at Oak Ridge National Lab. We've broken it out by actinides, others, and then the ones on the right are fairly new radioisotopes that we're bringing into the fold. So this just gives you a sense of the depth and breadth of what we have the capabilities to do. So covering some of our industrial isotopes, I've only picked out a few of them to highlight them. I was trying to find ones that I thought might resonate more with folks. So the first one is nickel-63. It has a very long half-life, as you can see, greater than 100 years. It also takes a significant amount of irradiation. So we start with enriched nickel-62. So nickel-62 does not typically in abundance in nature. So it has to be enriched. And this is going to come from previously at Y12, where we had the enrichment capabilities to make the nickel-62. But we then press it, and you can see this insert right here in this black square. We'll press the nickel-62 oxide into pellets. We will load them into these little capsules, which we affectionately will then load into what we call rabbits. You guys know in the nuclear world, we have all these great nicknames for things. So we'll press the pellets, load them into the rabbits, into the capsule, weld the ends, encapsulate that into a rabbit. And as you can see, it takes 15 cycles in the reactor. And that is just because the nickel-62 has a fairly decent but low cross-section. And cross-section just means the, what's the word I want? The probability that a neutron is going to interact with nickel-62 and can be transmuted into nickel-63. Has a fairly low cross-section, therefore it takes a lot of cycles. Remember what we said, it's 6 to 7 cycles a year. So if you do the math, we're looking at about 2+ years that these little capsules are in the reactor to meet sufficient quantities of the nickel-63. Nickel-63 is pretty much 100% beta emitter. So betas are like electrons as far as the size and energy. But the applications, if any of you have traveled through the airport and got sniffed, so you got pulled aside and hold out your hands, or they took a swipe and they took it over to the machine to be sniffed, it is a nickel-63 is the source that's being used for that detector system to do that sniffing for explosives or hazardous chemicals. Also, nickel-63 is being looked at for nuclear batteries, so using the beta voltaics capabilities for radio batteries. Also, if you notice in the lower picture, it is that beautiful green color. So as it is being processed and purified, it does come out to that beautiful green color. Selenium-75 is another one. It's got a 119-day, almost 120-day half-life. It starts with the irradiation of selenium-74. Again, a single neutron capture goes to selenium-75. So it has a high affinity for neutrons to transmutate, so it only needs one cycle in the reactor. It is a gamma emitter, but it's predominantly used for gamma radiography, a lot of times in pipelines and remote areas, so that they can bring out the radiography system and be able to use it in places that you couldn't bring out big equipment for. What you're seeing in that picture is actually a 3-bore hold capsule. It's kind of hard to see, but there are 3 tiny little holes that are welded in there, and we can load these are tiny little pellets into that capsule. And once it's irradiated, we take it to one of our hot cell facilities. We cut it open, pour out one of those holes at a time, inspect the little capsules, and then they get packaged into special form capsules and then transported to the customer. So we don't do any processing for the selenium-75. The promethium-147, this one's different. We actually don't irradiate for this particular version of promethium-147. In the future, we will be making it differently. It has a half-life of 2.62 years. Right now, we are harvesting it from the plutonium-238 production that we do on site. So plutonium-238 is made by irradiating neptunium-237. It does a couple of neutron captures, and then it beta decays into the plutonium. So the promethium is actually a fission product from that irradiation processing. So we can extract that, we can do chemical processing and extract it through that direction. It too is a beta emitter, It is used for beta thickness gauges for thin films, also being considered for nuclear batteries like the nickel 63, and it has been used in medical imaging. What you're seeing in this insert, it is a pretty pink color. Remember, nickel was nice and green. Promethium comes out kind of this rose pink color. And that is a picture of one of our hot glove box technicians actually doing the processing. So strontium 89 is another one of the isotopes. And you're going to kind of hear repetitive irradiation, neutron capture, chemical processing. So it's going to be a theme. Strontium 89 has a 50-day half-life. It starts by irradiating of enriched strontium 88. It too only needs one cycle in the reactor. It is brought back to building 4501 into the hot cell facility, and we'll do the chemical processing and separation and purification of that. It is used in bone pain palliation. The pictures you're seeing here are of actual irradiated pellets. And then you can see as you cut open the capsule, looking in on those pellets. On the left is just walking you through the flow sheet of the different type of processing steps to get to that purified strontium 89 product for shipment. Tungsten 188, as you see, has a half-life of not quite 70 days. It starts from enriched tungsten 186. So it's in the reactor for 2 cycles. It has to do 2 neutron captures to get to the tungsten 188. But what's interesting about the tungsten 188 is it's the K daughter. So the daughter is the rhenium. I always get this wrong. Did I get that right, Sandra? Okay. Rhenium 188, which as you can see has a very short half-life, less than 17 hours, but it has a high energy beta emitter. So we can load the purified tungsten onto a tiny resin package, and that is called a generator. And the generator is what's sent to the customer, so it can milk off the daughter product. So as the tungsten 188 decays to the rhenium 188, it's the rhenium that is used for the nuclear medical applications. And currently there are trials for the management of various tumors, the bone metastases, and rheumatoid arthritis. What you're seeing in our pictures here is those are rings. So instead of pellets, the tungsten 186 is made into rings, and the rings are then loaded into those little capsules that are irradiated in the reactor. When the rings come back, we run them through their processing steps to separate out the other products. Also, part of that is they can sometimes make osmium. So we have a gas trapping system inside the hot cell. So this is what the gas trapping system on the left looks like, all clean, shiny, new. This is what it looks like when you put it into the hot cell and you're looking through that leaded glass that's filled with shield material. So it kind of gives you that tint inside, but it still looks pretty good. You can still make out all the components of what we're doing. And then back to the far right on the inside here, you're seeing as we're putting those rings into that furnace to actually convert the pellets into and drive off the products that what we want and what we don't want. So switching gears, those kind of were some of the big industrial ones, but getting into the next level, the transuranic. So anything beyond element 92. And you can see our timeline here is, Fermi in the 1930s. We're still figuring out what is fission. We're still trying to figure out what a neutron is. All that's happening in this time frame. And then in the 40s and 50s, you have this explosion of identifying what the actinides are. And that was, Glenn Seaborg figuring that out and proposing the new row on the periodic table for the actinides. And then in the 1960s, we're pushing that envelope even further. And again, that is when the high flux isotope reactor and the Radiochemical Engineering Development Center, the Mountain Valley complex, is coming online. Because in order to make those transuranics or even transplutonium elements, you must have 2 things. You must have the starting material, but you also have to have that high flux, which is what the high flux isotope reactor gave us. So in the beginning of the program, we were actually starting with plutonium-239 material that came up from the Savannah River production reactors down in South Carolina. The targets were made out of that. And through going to the right on this chart, it's basically neutron captures. So you still stay within the same element, but you're upping the neutron. So you're creating isotopes. If you have this beta, your beta decay actually creates a proton. So it is kind of like the alchemist, but we're not creating gold from lead. We can only go up one element. So you can see something like the plutonium-243, when it beta decays, it goes up to americium-243. So through the series of neutron captures to the right and beta decays going up and to the left, that is how you can transmutate and get up to the curium isotopes, then to the berkelium, then to the californium, einsteinium, and fermium. Fermium is as far as we can go physics-wise in a reactor-produced situation. After that, you've got to go to linear accelerators. So how do we do that? Currently, we start with curium isotopes, predominantly the 246 and the 248, the heavier ones. We blend the curium oxide with aluminum. We press it into these tiny little pellets that you see here in the forceps. And we put about 33 of those pellets. If you look on the bottom, our pellets are not red, but if I use gray, you'd never be able to see them. So we fit about 30 to 33 pellets in our targets. Now I'm talking targets. These are full length targets, not rabbits that were about 4 inches in length. These targets are about 24 to 28 inches in length. So these are full length targets. The upper right insert shows you how you put the pellets, the spacers, fit them into the tube, and then we put a coolant shroud on them. The targets then go in. We have to first test everything with these targets. It's loads of fun and I must warn you, when we make these, we are doing this inside a hot cell. So we are pressing these tiny pellets, loading these targets, then compressing them, welding them, leak testing them, and putting the coolant shroud all remotely with manipulators. So to say it's a challenge is an understatement. To say that our folks aren't amazing, they are. To have the patience to do this. Once these targets are fabricated, we're going to then load them into the high flux isotope reactor. We load them into the center region, which is called the flux trap, which has that high 2.1 times 10 to the 15 neutrons per centimeter per square per second. And then we have to leave them in there for about 4 to 5 cycles. Remember, a cycle is about 23 days. After we're done with the full radiation, we need to let them cool for about 90 days to let the iodine-131 decay. And then we're typically looking at about 100 milligrams of californium-252 at the end of a radiation and about 10 milligrams of berkelium-249. And yes, I am really saying milligrams, not grams, but milligrams are what our campaigns look like. So then we got to process them to get to the final product, which is the wire sources that go out the door. So you're looking at 7920 versus 7930. That is a lot of processing. Again, everything done in our hot cell facilities, all done remotely with manipulators. So the targets come in, we dissolve the targets. We're going to run them through a whole bunch of chemistry that I'm not going to bore you with unless you want to know later on. I'll answer questions. But we have to go ahead and dissolve, get into a solution form, separate out the fission products, separate out the lanthanides, and purify the actinides. We also completely recycle any of the americium and curium because only about 15% is transmutated. We want to recycle the remaining and make it into our next set of pellets to go back into target fabrication. So we do have a full circle here. The products are our actinides, so the berkelium, californium, einsteinium, and fermium. We're going to purify them and run and separate them and run them through the different processing. We will do further processing and purification for the berkelium, einsteinium, and fermium. Those are used for research. So they're going to be used for radio and nuclear chemistry research, as well as super heavy elements, which I will talk about here in a moment. The californium is then going to go to our other hot cell facility where we run it through all of this chemistry and processing on the right. to make it into the wires. And why we want to do that is californium-252 is a very unique radioisotope, especially in the nuclear industry, because it has a relatively short half-life of 2.64 years, but 1 microgram, now I've gone from milligrams to micrograms, 1 microgram of californium-252 emits on the order of 2.3 million neutrons per second. So it is a portable neutron source, and it has a huge amount. I've only put a few up here for the applications. It has a global market, and there are only 2 places in the world that can make californium-252 on the scale necessary for the end users. And that is either Oak Ridge National Lab or in Russia in Dmitrograd at the SM reactor. But the applications within the energy sector, oil exploration, it is used for oil or for well logging. So the neutron source can go in, it pings back a signal, and that signal can let you know if you've got hydrogenous, i.e. either water or oil deposits. Nuclear fuel rod scanning. So the neutrons are used as you're scanning the full length of the fuel rods, looking for the homogeneity of the uranium-235. Those thermal neutrons will interact with the uranium-235, give off other neutrons, and by measuring those, you can know what that enrichment looks like. It is used for reactor startup sources for any new reactors or reactors that have been shut down for a significant amount of time. Those neutrons can be used mainly to show the instrumentation that, hey, there's neutrons there. It can also help start with the getting to the critical state. Coal analyzers, and this is also true in infrastructure. The coal, mineral, and cement analyzers are used such that you use 2 neutron sources of similar strength on either side of the conveyor systems, such that they are activating any impurities. Those impurities give off gammas, those gammas are detected, and based on those energies, you know what the impurities are, and you can move the stream as you need it to be. It's also used for radiography, similar to gamma radiography. You can do neutron radiography, and that's been used for large castings, turbine blades, landing gear on jets. So it has a broad application. Also can be used for measurements for corrosion on bridges, highway, and infrastructure. So again, incredible isotope. If you want to know more, that's been my baby for 15 years. I can answer any questions on 252. Switching gears again, going back to the super heavy element discoveries. So remember, we're kind of all things actinides. So you can see that a lot of the targets that were used for the discovery of these different super heavy elements, a super heavy element is any element greater than 103. That's the definition. So you can see that many of these Oak Ridge National Labs supplied the actinide target material for those discoveries. So why will we care about going into super heavy elements? Well, there's this theory. And again, Glenn Seaborg is kind of, the father of actinides. He was really the driving force for getting Oak Ridge National Lab to have the high flux isotope reactor and the Radiochemical Engineering Development Center. This is his theory of this idea of an island of stability somewhere out there in the super heavy element on the chart, the periodic chart or of elements. There could be elements that if you had the right number of protons and neutrons are stable. So why would we care? Why does that matter? Well, back in the late 40s, when americium was discovered, it was a very short-lived isotope of americium, so no one knew what to do with it. Most smoke detectors in your homes have a small americium-241 source in them. So we don't know until we can find these that last longer, what their properties might be and how we might be able to use them. So we truly are in an area of discovery. We just don't know. That's the cool thing right now. So what are the next steps? So there is no one country right now that can do it all. It must be a collaborative effort. The United States, we are the ones that are making the actinides in sufficient quantity that can be used for the targets. And these 3 facilities in Japan, Germany, and Russia have the accelerators that are necessary to bombard those targets to push up to the next super heavy elements. So how we make super heavy elements is you have a target wheel that has the actinide, whether it's curium, californium, berkelium, and that is spinning, and then it is being hit by a beam that's been accelerated, and the beams have typically been of calcium 48 ions, but now these facilities are building these new accelerators such that they could accelerate beams of higher than calcium. So vanadium, titanium, possibly even iron, seems insane, but higher, higher mass, higher intensity, higher energy, so that they can hit these targets and push up beyond element 118. Japan is currently using curium from Oak Ridge National Lab in a beam of vanadium looking for element 119. They have been making upgrades to their accelerator to have higher energy and current. The Emperor has made that a focus of their science program. Germany doing the same thing. They are upgrading to a new linear accelerator called a HELIAC for greater beam intensity, but they are also looking at performing chemistry on the super heavy elements as they make them to understand their properties and do they really fit on the table the way we think they should. In Dubna, Russia, they have also built a brand new super heavy element factory. And so they are pushing to look for those heavier isotopes of element 118, which is oganesson, and see if they're getting close on the island of stability, but they will also be pushing for 119, 120, and beyond. So that's kind of a flavor of what we do and what's going on right now. We want to make sure that we thank the Department of Energy with respect to the Isotope Program Office, which is managed by the Office of Science, the High Flux Isotope Reactor, which is a Office of Science user facility. Oak Ridge is managed by the UT Patel contractor, and several of these folks have helped in getting this presentation put together, and I wouldn't have been here if it wasn't for all their work. And this is what the periodic table looks like today. And hopefully here shortly, we will figure out whether or not we have 119 under column 1, or do we have a whole new line? And are these things where we really think they should be? So with that, I am done, and I'll switch it back to Stevie.
All right, I'm unmuted. Okay, thank you so much, Julie. It was an excellent presentation. So now we will hear from Sandra Davern.
I'm just sharing my screen.
Yes, you are.
Bear with me for a moment. Okay, can everybody see my slides?
Yes.
And I'm in presentation mode.
Yes, you are.
Okay, awesome. Well, hello everybody, and since I'm not sure what time of day it is, welcome, and I'm really grateful for this opportunity to speak to you today. Thank you to the organizers of USWIN. This is a privilege, and I really appreciate that I get to tell you this wonderful story of medical radioisotope production at ORNL and what moves we're making in radiotherapeutic development. So tying into what Julie has already mentioned, we have a long history of making medical radioisotopes here at the lab that continues today. And along the way, the lab has been involved in radiobiology, looking at the effects of radiation on cells, developing isotopes specifically for treating cancer. You can see, I'm looking for my pointer, by the way. You can see RusNap here has a little generator, as Julie mentioned, which is basically a resin this is a tungsten rhenium generator that Julie has talked about for cancer and heart disease treatment. Rose Bowl and Saad Mirzadeh have both been very instrumental in pushing forward actinium-225 and its decay daughter product bismuth-213 for radioisotope therapy as well. And today we are actually the critical raw material producer for actinium-227, which is important because it is the material that makes radium-223. And as I will explain to you, that is currently the only FDA-approved alpha-emitting radionuclide used in cancer treatment. So I'm going to talk a lot about something called targeted radionuclide therapy. And just to give you an idea of what that involves, it is a very multidisciplinary field. It involves everybody from chemists to biologists to nuclear engineers and all along the way. So the whole premise of this is that you have a radionuclide, be it an alpha emitter, a beta emitter, or an Auger emitter, that is caged in what we term a molecular construct. So some sort of a chelator or a nano construct, a nanoparticle. It's held and attached onto a targeting molecule via this red linker here. And the targeting molecule can be anything from a small molecule, a chemical compound, it could be a peptide, it can be something called a monoclonal antibody, or a fragment of a monoclonal antibody. And what I want to highlight here to you today is the differences between alpha, beta, and Auger electrons, which basically, as you can see in this image on the right here, stems from their radius of effect within a cellular environment. So you can see here that the alphas have this radius of effect, which is about what we would term between 50 and 100 microns in distance. And just to put that in perspective, a human hair is approximately 70 microns in diameter. So you're traversing not much more than a human hair when you're emitting alpha particles. Auger electron emitters need to be delivered inside the cell, right up against the nucleus of the cell to exert their effects. And then beta emitters have a much larger range on the order of millimeters. So quite a few more cells. Targeted radionuclide therapy has a lot of advantages because, as you can see in that cage that holds the radionuclide, you can swap in and out different therapy or imaging radionuclides to suit your purpose, tethered to that same biological targeting molecule. So you can actually visualize where your radionuclide should go to exert that therapy by using an imaging agent. And that is really one of the beauties of this therapy, that and the fact that it precisely delivers your radiation where you want it to go. And you don't have the same thing that you do with chemotherapy, where it's just systemically delivered and goes wherever it wants. This is targeted therapy. So you're delivering your radionuclide to the cancer cells where they can maximally deliver their effective dose and then minimize that effect on normal surrounding tissue. Here's some images so you can see when you use that imaging agent, what you can do with what the power of this kind of therapy is. And in this instance, we're looking at metastatic prostate cancer. There are a range of patients here, and in red, you can see how each of these little red dots actually correspond to metastatic cancers that are in this particular individual here in the bones of that patient, because he's got prostate cancer, and the prostate cancer has metastasized. And normally when that metastasizes, the bone is where it likes to go. So you can see that this patient here has been treated with lutetium-177, which is a beta emitter, and there's quite effective treatment here when you do that. I will mention that in this study here on the right, I'll start down on the bottom, because in the bottom you have the lutetium treatment, which for this particular individual didn't work very well. However, when they came back in with actinium-225 treatment, now actinium is that alpha emitter with the shorter range. And what I did forget to mention to you is that actinium deposits a lot more energy in its, the alpha particle blows through the cell. It's like a little bomb going off. It's much more energetic than the beta particles. So your DNA in your cell is double-stranded. So you remember your high school biology, it's an alpha helix, there's 2 strands of DNA, and your cells can repair that damage, because we're all exposed to damage every day, and your normal cellular process repairs that. Cells are much more efficient at repairing single-stranded DNA than they are at repairing double-stranded DNA. So when you look at lutetium is a beta emitter, and it mostly causes single-stranded DNA breaks. Now, in a lot of these instances, as you can see on the left, that's enough. It's enough to cure and get rid of the cancer. However, in this particular patient, it looks like a resistance mechanism has developed. So now you can use your double-stranded DNA break, actinium-225, and in this instance, this seems to be much more effective at reducing cancer that has basically no other way of treating it. So this is the power of this therapy. What I want to show you now is that this is the theranostics approach, as I've mentioned. You can image and you can treat using different radionuclides. And sometimes you have radionuclides that can do both at the same time, such as the scandium-47, the copper-67, and the different rhenium isotopes we have here. So these different isotopes are all beta emitters, so they have that therapeutic effect, but they also have gamma energies that are useful for imaging. And spectral PET imaging is used to visualize the cancer within the patient at the beginning, before the initiation of treatment, and also at the end to see how treatment is progressing. So this is sort of the hot topic of isotopes for medicine. Mostly what has been approved have been beta emitters, and you can see here at the end, these 2 that I've bolded out are Lutathera, which was approved in 2018. It was the first sort of peptide delivered beta emitter because it's attached to this small molecule, which is a small peptide that recognizes specific receptors on neuroendocrine tumors. More recently, this Pluvicto has also been approved in 2021, and that's been attached to the target for prostate cancer, which is prostate-specific membrane antigen. The only alpha emitter, as I alluded to earlier, is radium-223. That was approved about 2013, and its brand name is Xofigo, but it's really radium-223 dichloride. So there's no biological targeting involved here. There is what's called self-targeting, because radium behaves like calcium and it homes to the bone. So this is an interesting process where you give the Xofigo, it goes to the bone, and it helps to reduce those painful bone metastasis that you've already seen in the prostate cancer patient. Actinium-227 production is what I've already mentioned, and we do that at Oak Ridge to provide what's known as the critical raw material for the active pharmaceutical ingredient radium-223. And you can see in this decay chain here that the actinium-227 has a relatively long half-life, almost 22 years, and decays to the radium-223, which is a short half-life of about 11 days. That makes it suitable for therapeutic applications. I'm going to have to go rather quickly through my slides because I realize I have a lot. But here's a sampling of the medical isotopes that we produce at Oak Ridge National Lab. I will talk some about actinium-225 in a little bit. I've mentioned 227 with its associated K-daughters, radium-223. There's another one, thorium-227, which also has potential as an alpha emitter for treating cancer. Radium-224 with lead-212, and tungsten rhenium and strontium that Julie has already referred to. So let's talk a little bit about radium. Radium's very interesting. So radium-226 has a very long half-life, and we irradiate it in HIFER to produce the actinium-227 that I mentioned, so that we can get the required radium-223 for that only FDA-approved alpha-emitting radionuclide. that makes Xofigo. However, a byproduct of this is thorium-228, and that has more recently been noticed as being attractive as a producer of some other radionuclides, which are also favorable for therapy. And then you can also go all the way across to thorium-229 so that you can get actinium-225. And you can see here on the right, one of our Andrew Burgoyne, who's one of our staff scientists here, is also an amateur photographer, and he took these wonderful images. He's done them for many of our isotopes, but this is showing you Cherenkov radiation. So you can see this nice blue glow that shows you about 12.5 millicuries of thorium-228 in a vial ready for shipment. So we have to thank Madame Curie for radium-226. It has a very high specific half-life. And I just want to highlight that what's interesting is that we're now producing a range of isotopes for medical applications from radium-226. But originally, they actually did use radium-226 in what's known as brachytherapy. And if you look down here on the right, these black sort of nail-like structures are what are known as medical devices that were implanted into the breast tissue as a way to treat breast cancer. I kind of look at it today and I think it's pretty crude and looks kind of barbaric, but you know, it's what they had. And now I will tell you a story about how we're recycling these medical devices to make the next generation of these radionuclide-based therapies to treat cancer. So we are extracting that radium-226 from these historical devices, and you can see them here. They are sort of nail-like structures or plaques, and they were implanted in people to treat cancers. So needles, we call them needles and tubes, but they're, you know, have a lot of different metals in them. So the separations chemistry is quite complex, but we do dissolve them up, and you can see them here. They get chopped up, they get dissolved, and then, you know, the radium gets extracted and goes through a number of different steps, and you precipitate a nice radium carbonate solution that is pure and can be used in the next step of the process, which is the production of targets that get irradiated in the high flux isotope reactor, as Julie has shown you. So we have Karen and David here working at this glove box, and you can see here, this is actually, I have to show you, this is the first pellet of radium-226 that was ever made at Oak Ridge National Lab. And I was lucky enough to be involved in this project. And it's really exciting to see when these things come about and you're making sort of first in your science. This is a couple of the pellets together, so they're being analyzed. And you can see here with that forceps, they're being loaded into one of those rabbits that Julie mentioned. And this device here is actually a welder that is located within a hot cell. So you can see every time you see that little kind of yellowy glow, you know you're behind the leaded glass, or you're looking through the leaded glass and looking into a hot cell. So the rabbits get welded, and you can see here, it's held in place. This what's called a little TIG weld in the end of the rabbit, and it gets an identifying mark so that we know where it is. But it's put in the high flux isotope reactor and irradiated. And as we mentioned before, depending on how long you irradiated, the irradiation times here on the bottom of this chart give you the different products that you need. But of course, there's a lot of chemical separations involved in helping you do that. So thorium-228 leads to radium-224 in its decay chain. And I will tell you a little bit now about our next medical isotope, which is radium-224 produced lead-212. Now, lead-212 itself is actually pure beta emitter, but people talk about it in terms of targeted alpha therapy. And the reason for that is that lead-212 decays, I'll go back, it decays to bismuth-212 down here. Bismuth-212 has about a 60-minute half-life. Lead-212 has a 10-hour half-life. So if you trap the lead-212 in that cage that I showed you earlier and attach it to your targeting vector, you can deliver the lead where you want it to go to the tumor cells, and then it will spit out the bismuth-212, which is the alpha emitter. So you're actually getting alpha and beta decays at that site. But we've seen that the alpha is a more powerful therapy. And this is known as what's also known as an in vivo generator. So you are generating that bismuth-212 continually from that lead-212. And on the left, you can see what it looks like when you make the little generator for shipment out to our customers, with which the generator is the radium-224 to produce the lead-212. And if you have any questions, we can go through that later. This is one that's ready to ship. Just going to highlight the last one in line here is thorium-229. That's really important because ORNL actually at the moment has one of the main supplies of thorium-229 for the production of actinium-225. This is another generator-derived radionuclide. And a generator just means that you have chromatography resin, like you see here in this shielded hot cell cave. This big column is full of resin, and you're actually looking at what we call the actinium-225 cow. So it's thorium-229 is loaded on this resin, and you can wash off that actinium-225, which is your medically important radionuclide. So I'm not going to go through it because we don't really have time, but there are a lot of different processes that are critical to getting the pure radionuclide that you want, so that it's suitable for medical applications, because you want to remove all the trace impurities. I just want to highlight too that when you look at this decay chain, there are multiple alpha decays in the decay chain from actinium-225, which has approximately a 10-day half-life. So I'm going to talk a little bit now about how we think about these radionuclides that we produce and how we want to best sort of cage them so that we maximize that therapeutic effect and minimize our side effects. This is what I get excited about. I get excited about the other stuff, but I'm probably a bit more excited about this. So I want to look at what are the cellular impacts of these targeted radionuclide therapies. And I've shown you images of what's mainly been done so far has been related to prostate cancer. Actinium-225, just so you know, there are about 23 clinical trials ongoing right now in the US, looking at how we can use actinium-225 to treat various different types of cancers. And how you treat various different types of cancers is you switch out that biological targeting component and you hit the cancer that you're interested in. But this is new therapy, and so there's a lot of questions that need to be considered, especially when you're thinking about an alpha emitter, actinium-225, that has such a huge energy. That energy can actually break the chelate if it's just a coordinated chemical bond that holds it. So you want to get it where you want it to stay quickly, because you don't want these decay alpha daughters. Now, the main ones to consider here are bismuth-213 and francium-221, because if they relocate from the cancer site to normal tissue, they can have adverse effects there. And I'm highlighting a question here that I like to think about is, well, what about radioresistance? How do we start thinking about that? So we can make nanoparticles. We have a couple of different ways of doing this, but this particular one is using lanthanide phosphate salts. So you're basically encasing your actinium in a nanocrystal. And that nanocrystal, we're attaching it to an antibody, in this case, something that recognizes a receptor specifically on lung cells. And you can see in this mouse image here that, yes, when you inject it into the tail vein of a mouse, it specifically goes to the lung, and it's not going anywhere else. So this is kind of the beauty of this. Nanoparticles also have the ability to give you what we call those in vivo generators. So you have the actinium-225, it's constantly decaying to its decay daughters, but by keeping the actinium-225 within a nanoparticle, you are also retaining those decay daughters. So you're keeping them where you want them to go. At Oak Ridge, we've lost the ability to do experiments in mice. So we have been developing more advanced, physiologically relevant biological systems that can use human cells. This is an example of a microfluidic device that we manufactured ourselves. And some of the advantages of using these types of approaches for studies is that you can screen without animal testing. You can use different single cell or multi-cell microenvironment complex studies of multiple cells. And you can do studies in situ on the chip where, like you see here, we're staining within the channels so we can see our cells. And over here you can see, I have better pictures in a minute, the effects of external beam irradiation. So all of these little green dots within the blue are DNA damage that we have induced with X-ray irradiation in this instance. I'm giving you a flavor of what we can do with the biology so that you can see how we're thinking about looking at the effects of these radionuclides. On the left here, these are what I'm calling mini tumors. So we can make in dishes these little, spheroids that are breast cancer cells. This is a confocal microscope image here on the left. So all of these little circles that you see are actually slices through a spheroid. So it's a round, clump of cells, but we can slice all the way through it and see. The blue dots are the nucleus of the cells, and the red are our nanoparticles that I just talked about. So we can see that these nanoparticles, even if we don't target them, get uptaken into those tumor cells. And this has a little fluorescent dye on it so we can see where it goes. And our next step is to look at, well, how efficiently does it shrink and get rid of those tumor spheroids when we have our radionuclides in there. In this little picture here, I'm showing you what an alpha track through the nucleus looks like. So an alpha particle will go in a linear direction and will deposit all of its energy in that linear direction. And you can see in this cell here that each of these little white dots corresponds to that DNA double strand damage that I mentioned earlier. And in this particular instance, we're not even targeting. So this is what happens when we just put free actinium-225 in association with cells. So we're not targeting it, we're not making it go to the cell, but when it randomly hits, you're seeing that we're getting DNA damage. I have a little movie so that you can see that we have an ability to do live cell imaging. So the blue that you're seeing is actually the uptake of some of our nanoparticles within the cells. And I'll mention that these cells are patient-derived ovarian cancer cells. And you can see down here, this scale bar is 200 microns. So I looked at it earlier, and I think 100 microns is about 5 of these cells. So if we're thinking about that alpha emitter going no more than 100 microns, it'll smack through about 5 cells. And, hitting the nucleus, you're really going to get the bang for your buck there. And these are unpublished results. This is relatively recent. So I'm almost done. This is just a reminder. Let's see if you can remember anything I've mentioned as I've blown through this story of radioisotope production for medical applications and what we're trying to do for developing new radiotherapeutic interventions. I've mentioned that we have alpha particles that have this huge energy to break the 2 strands of DNA. I've mentioned that the beta particles can also induce single strand DNA breaks, but they have a longer range in tissue. I didn't talk a whole lot about Auger emitters, but they are actually as powerful as the alphas, but they need to be closely associated with the DNA. And so they can cause double strand DNA breaks as well. This is a different representation of that targeted radionuclide therapy. Again, you have your biovector, which is your targeting molecule, linker. And in this instance, we have a little fiddler crab holding on to our radioisotope. So this is sort of our representation of that chelate or the nanoparticle, something that's holding on tight to what we want to deliver. And, we're looking at these therapies being injected into patients, distributing throughout the body, homing or localizing to where the cancers are located, being retained there so that you're creating the maximum cytotoxic effect where you want it to be, not systemically. So targeted radionuclide therapy, as I've mentioned here on the bottom, is really good for both localized and metastasized cancer. And it needs to be added into the surgical brachytherapy, external beam irradiation, and chemotherapy approaches that we have today. And so I like this image because it shows how therapeutic interventions for cancer have expanded over the years. I know this is 2015, but you can see in this image that these are all the different cells that are involved in a tumor. It's not just the tumor cells, there's a whole load of accessory and immune cells involved. And I'm sure a lot of you have heard about immunotherapies, checkpoint inhibitors. So these have really come in vogue lately, and they're very, very good. On top of the chemotherapies, and we have, radiotherapy is mostly still seen as external beam, single or fractionated dose. My question is, where are targeted radiotherapeutics or targeted radiopharmaceuticals? And what we want to do is push this therapy further back in the intervention process, because we do believe it will be much more effective if we can use it sooner. Most of the studies with clinical trials and what's approved to date are for palliative care or for end-stage disease. So being allowed to administer this therapy earlier and deciding on how to integrate it, what are the standards of care, are really the next frontier in how we move this therapy forward. I'm done with my presentation, but there are some opportunities at Oak Ridge National Lab. The Glenn T. Seaborg initiatives are, Seaborg was interested in training the next generation of researchers in transactinium science. And really this relates to both what I'm researching and what Julie's doing. The education of young people is so important, and we really value that at ORNL as well. And there are 5 labs that have Glenn T. Seaborg institutes, and ORNL's was just established last year. So if you're interested, our director is Samantha Shrell. And the goals are to maintain and enhance the capabilities of the US in actinide science and technology, and to build that community of actinide scientists at ORNL so that we can, bring with us that next generation of innovative thinkers and contributors to medical radioisotope research and finding that island of stability, many other applications as well. I just want to thank our sponsors for some of this work was sponsored by DOE Basic Energy Sciences, by the Isotope program. I have to thank the Oak Ridge National Lab for the lab-directed research and development that has funded a lot of this, and everyone in our accelerating radiotherapeutic through advanced molecular contact. We call it the ARM initiative because it's a mouthful. And the ARM initiative because we're arming our biological targeting vectors with that radionuclide, so with our radioactive payload. So again, thank you, and I'd be very happy to take any questions.
Thank you, Sandra. We now will open it up to the audience for questions. Please remember to raise your hand if you'd like to be unmuted, and you can ask your question yourself. Otherwise, please feel free to type your questions into the chat box. And we actually already do have 2 questions. So I'm going to go ahead and ask these for Patricia Valdez. Does the Russian lab also do R&D for medical purposes? Because I have not heard of Russian cancer treatments. And then number 2, what quality assurance standard is used in producing medical isotopes for medical uses?
So I think Julie should take the first one.
Finding out what the Russians are doing is incredibly challenging. The open literature is not easy to find. You've got to go digging pretty hard. So I can't say affirmative, but I would have to say they must be doing something. They have the capabilities to be doing that. But as I said, it's just not as easy to find their work in open literature.
Yeah. And for medical isotope production, you need what's called current good manufacturing practice, because that is the FDA's and the world regulatory bodies standards that need to be met to make sure that you're not injecting a patient with something that is not what you think it is. So you want to make sure there's no contaminants in there. There's nothing that's going to have adverse effects apart from what you know the medicine is going to do to them.
Okay, we do have another question from Tamara, our chair. What are the impacts of Russia's invasion on Ukraine?
Challenging.
Yes.
Because of that. So, you know, one of the things is in the past, we've always done collaborations with the Russians, especially in the super heavy element world. And that was even done during the Cold War. But now the State Department has stopped. We cannot do any scientific collaborations with Russia. So that was painful. And also, many countries have chosen not to procure their radioisotopes or even their stable isotopes from Russian providers. So they're looking for alternatives. And again, like we said, there's only 2 of these types of reactors, these research reactors that can make a lot of these radioisotopes. So that has put more, I don't want to say stress, more opportunities for Oak Ridge National Laboratory. So it's been keeping us busy. So if anybody's interested in, like Sandra said, jobs.ornl.gov,
Yeah.
we are looking for more bodies to come support this effort.
Okay. Let's see, I've received a few more questions before the presentation. So where do you see radioisotope research and development headed? So what do you think is next?
That's a big question. I obviously think that there's a lot to be done in terms of medical radioisotope research and development. You know, how do we combine radionuclides for more targeted effects that have both long range and short range, alpha and beta emitter capabilities? And how do you combine those with potentially chemotherapy drugs for more precision medicine? So that you're minimizing side effects all the time. You're thinking about maximizing your effect to the tumor and minimizing side effects. That's one particular big area. Another is understanding the fundamental properties of these isotopes, and that's part of our initiative too, is getting to know what the crystal structure of some of these radionuclides are, because not many people can do that research. We can do it because we have the instruments and the equipment that you can put a highly radioactive crystal of, you know, one of the radionuclides on a single crystal X or D and, you know, figure out what that crystal structure is. So these are some of the types of experiments we are looking at, and that information then helps you understand the coordination chemistry around that radionuclide so you can better design that molecular cage to hold on to it. So it's all additive.
Okay.
And then Julie, I'm sure, has more.
Well, then there's also just the basic research, right, with the super heavy elements, trying to figure out where does the periodic table end? Where would it end? How far does it keep going? From that, you know, understanding the chemistry of these super heavy elements. Again, the questions are coming up now as they can literally do atom by atom chemistry. So some of the isotope or elements like 108, 109 that live a little longer, there's doing chemistry on them, and they're finding out that maybe they're not where they're supposed to be on the periodic table. So that could shake up the whole periodic table, at least with the newer discovered elements and our understanding. So what Sandra was saying with the coordination chemistry, there's a lot of that going on with the heavier actinides, not the super heavies, but the berkelium, the californium, and if we could ever make enough einsteinium, they would be wanting to look at that to really understand the physics and the chemistry behind these. So there's that kind of basic research, but then more on the applied and production side is, how can we do this better? We need to make these radioisotopes, whether it's for industrial or medical applications. How do we do the chemistry better? How are we more effective? How can we make more how do we make more with less? How many times have we all heard that?
Yeah.
Same thing in our world of, how, if we can be more efficient, more effective, then we can get things out quicker, faster, cheaper into the world for their use. So there is a lot of work, and we have an amazing Department of Energy sponsor within the Isotope program who wants to see all this happen. So we are growing and really need as many folks coming in as possible.
Well, that's a perfect segue to this next question. So what degrees or skills do you need to go into radioisotope production?
Everything.
So true.
And the reason I say that is we are maintaining hot cell facilities, glove boxes, a reactor. All of those are going to need craft. So we need the skill of the craft. We need millwrights, pipe fitters, electricians, instrument and control technicians. We need mechanical engineers. We need chemical engineers. So, we need technicians. So basic chemistry background, 2-year degrees in associate's degrees within chemistry, mechanical systems, even like with Sandra with biological, we can teach you the radiological side of things.
Yep.
But from my perspective as applied in production, I'm not kidding when I basically say almost everything. We even need computer science. We need to do the modeling and simulation for how to make these. So,
Right.
and then Sandra's going to take the research side.
Oh, no, but you need the nuclear engineers to figure out, theoretically what you want to put into the reactor, because you need to do all that theory before you put anything in there, and you need to meet the requirements of HIFR for safety basis. So that's a huge thing. The safety basis for everything that goes in the reactor is huge. And that's a whole quality assurance side to that as well. And then, of course, there's quality assurance based on the products that you're producing for shipping out. And quality at different levels. There's quality for what's going out to make a nuclear battery. That's one level of quality for your customer. But then there's quality for those medical applications, which is a different, more stringent level of quality as well. So you have that gamut of backgrounds of. And I think that's why I love it, because we have so many different people with different diverse ideas and specialities. So we have chemists, separation chemists, inorganic chemists, organic chemists, biochemists. If it's got chemist on the end, you're pretty good. It's got engineer on the end, you're pretty good. It's got an associate's degree. So there's a lot.
And the other maybe technical side of things that we are growing is our financial side. Our financial officers who help us track the cost of these programs and do our monthly spend plans, project controls to keep track of our schedules and our resources, because we're all competing for those same resources. So it's not just the technical side. We've also got the business side that we need support with.
Excellent. Yeah, every person you mentioned, I've worked with at a nuclear power plant. So,
Yeah.
Anybody's interested, they have some excellent opportunities. So just looking through the chat, there is I do want to read this because I think this is worthwhile reading. A hat tip to US Wind for sharing about these targeted therapies back in 2015 at the national conference. Julie, you facilitated that session. So that's from Kara Lucardit, I believe at Duke. So just looking through the list, I don't see anybody with their hand raised. So this is your opportunity if you have any questions you'd like to ask. Okay, I'm not seeing anything. So again, I would like to thank our panelists, Julie Ezold and Sandra Davern, for their excellent presentations today. I learned a lot. I'm sure everyone else did from this discussion. So I will now turn this over to Tamra for her closing remarks. Thank you. You're muted, Tamra.
I would like to reiterate that. I really want to thank Julie and Sandra again for sharing such insightful information today, and Stevie for moderating the program. As I mentioned at the beginning, I have a business background, so I love learning something new and outside of my area of expertise, and I just suck all of this in. Thank you so much. And Stevie, you did an excellent job moderating the program. I also want to recognize Eunice Derez and Shantika Glover, and they are US Wind's Professional Development Amplify Subcommittee co-chairs. And so they have helped us beginning in February and throughout the month of March, promoting all of the events we've had, as I mentioned in the beginning, several webinars this month and some other things going on to celebrate a Women's History Month. And we couldn't have done it without their help in promoting on social media and just getting the word out. So I really want to recognize all of their help. And finally, I want to thank all of you who attended today and took the time out of your busy schedules to join in this conversation and help us celebrate the women who tell our stories. I know it's difficult taking time out of your busy day to work in professional development and learning opportunities, but it's key to our profession because, as we always talk about, our journey to excellence just never ends. It's ongoing, and it's with these opportunities we're able to just be more successful in our jobs, our careers. And thank you on behalf of the Professional Development Committee for allowing us into your day. Also, if you are joining today as a group, please send your chapter name and the number of people in the group to uswinpd@gmail.com, or you can also drop that information into the chat so we know how many people are actually attending outside of those people that are actually have their names typed in today. And finally, another thing that's key to nuclear is feedback. We all know that feedback's a gift. So after you leave today's webinar, a survey should automatically pop up. Please take a few minutes just to complete it so we know how we're doing, if we're meeting your needs. And if the survey doesn't pop up, please just check your email. You should have it there. But we do ask for that information, and it's how we can continue to provide you with the programming that you are looking forward to. And with that, thank you all again and have a great week.